Good afternoon. My name is Elizabeth McKenzie, and I'm going to be talking about mitochondrial fatty acid beta oxidation disorders. Basically, these disorders affect the ability of the body to oxidize fatty acids to produce acetyl-CoA, which is going to be used in the Krebs cycle to produce ATP. So, beta oxidation has to do with the production of energy. It's a multi-step process, so as you can imagine, there are lots of points along the way um, and for glitches in the system, so to speak. And we're going to be focusing on two of those. But just as a general overview of where we're going to be going in this presentation, I'd like to provide a little bit of a background of fatty acid oxidation disorders. We're going to be looking at a review of the beta oxidation process and all the steps that are involved with that. Then we're going to be looking a little bit of an overview of what certain fatty acid oxidation disorders are, and then we're going to be focusing on two specific ones that have to do with carnitine palmitoyltransferases 1 and 2. And then we're going to be looking at the association of CPT1 and 2 disorders with type 2 diabetes. So, to provide a bit of background, fatty acid oxidation disorders are autosomal recessive, and these disorders are really significant for the individual who has them because that affects the energy production and it can result in some really serious consequences health-wise. There are two different groups of fatty acids. There's the short and medium chain fatty acids. These guys are transported directly into the mitochondrial cytosol. However, the long chain fatty acids, the ones we're going to be talking a lot about today, have to be conjugated to carnitine in order to be transported across the mitochondrial membrane and then released as acetyl-CoA to be used in beta oxidation pathways. As a review of the beta oxidation cycle, we have our fatty acid synthesis that's going to occur in the cytoplasm, but the oxidation, which is going to occur in the mitochondria. So the first step is going to be taking our fatty acids from the adipose cells to transfer them to other cells in the body to be used as energy. So those fatty acids have to first be cleaved from their glycerol backbone via an enzyme called lipase. So when lipase cleaves them, that means that fatty acids are free to travel from the adipose cells through the bloodstream to the particular target cells. Fatty acids are going to enter the target cells via a transporter, a fatty acid transporter, and this transport is necessary because the cell membrane is negatively charged. Fatty acids are then activated via acyl-CoA synthetase, ATP and CoA, to form a molecule that's called acyl-CoA. Now, acyl-CoA is transported through the outer mitochondrial membrane via porins. Acyl-CoA is then going to enter the inner membrane space and then cross the inner mitochondrial membrane. However, it can't just cross because the inner mitochondrial membrane is actually impermeable to acyl-CoA. So acyl-CoA is going to have to utilize something called the carnitine shuttle. Acyl-CoA is charged with carnitine via carnitine palmitoyltransferase 1, and then that CoA, which is attached to it, to the acyl group, is going to be recycled to be used with the next fatty acid that comes along. But anyway, with the result of that cleavage is called acyl carnitine. So here's a better view of what the carnitine shuttle looks like. So acyl carnitine is going to be transported across the inner mitochondrial membrane via the acyl carnitine translocase. But in order to regenerate acyl-CoA, which is what we need, it has to utilize something called the carnitine palmitoyltransferase 2 enzyme. And what this guy does is reattach our CoA to the acyl group and then take the carnitine that was on there and recycle it for the next fatty acid that comes through. So now that we have our fatty acyl-CoA inside a mitochondrial matrix, we're going to be able to continue beta oxidation. Now there are 4 general steps with beta oxidation. The first is okay, there's a little glitch in that. So the first step is dehydrogenation. So Here, our fatty acyl-CoA is going to undergo dehydrogenation by acyl-CoA dehydrogenase and to form something called enoyl-CoA. The FAD that's used in this as a reducing agent is going to result in FADH2, and FADH2 is what's used to produce ATP in the electron transport chain. So this is kind of the first way that energy is generated through the pathway. Now, the second step in beta oxidation is hydration. Here, our intermediate enoyl-CoA is hydrated by enoyl-CoA hydratase to form beta hydroxyl-CoA. Now, beta hydroxyl-CoA is then going to be dehydrogenated by beta hydroxyacyl-CoA dehydrogenase to yield beta ketoacyl-CoA. NAD is the reducing agent here, and NADH2 is going to be used in the electron transport chain. So the fourth and final step of beta oxidation is beta ketoacyl-CoA being cleaved by acyl-CoA acyltransferase, otherwise known as sialase. So our CoA-SH is going to be added, and the result is acetyl-CoA, and that acetyl-CoA is then able to go through the Krebs cycle, and that's where we get the large amount of ATP that the body needs to function. So at the bottom of the figure on the right, you're going to see that there are two products actually. There's an acetyl-CoA, and then there's some sort of fatty acyl-CoA. And the fatty part can be replaced with any sort of word like palmitoyl, for instance, palmitoyl acyl-CoA. So that product is going to undergo beta oxidation again, the same 4 steps as above, until it's broken down completely to acetyl-CoA. So as you can see, there are a lot of different steps in beta oxidation, so there are a lot of different opportunities for things to go wrong. And when things go wrong, we get something called fatty acid oxidation disorders. Now, there are 4 categories of them, but the one that I'm going to be focusing on the most has to do with the intramitochondrial beta oxidation defects. And those are going to have a lot to do with the carnitine palmitoyltransferases 1 and 2. So the attachment of carnitine to our fatty acid and then the recycling of carnitine to be used again. For all of these fatty acid oxidation disorders, they can be diagnosed at 3 different stages. There's prenatal diagnosis, newborn diagnosis through newborn screening, and then there is a diagnosis in children and adults. Now, in prenatal diagnosis, one of the key parts of that is actually mutation analysis. And this is going to be really important, especially if the mom has a history of liver disease. And that's going to be important, as I'll show in the next slide when we talk about kind of the physical manifestations or the phenotypes that result from mutations. In newborns, there's a newborn screen. They're going to be looking for acylcarnitine profiling using blood and mass spec, which is actually really cool. And then there is diagnosis in children and adults. Again, this is going to be looking for mutations in specific genes. They're also going to be looking at fatty acid transport studies because these are going to show what are going to show potential fatty acid transporter defects. So this is an outline of, on the left, general manifestations of those with fatty acid oxidation disorders, but then those that are more specific. So generally speaking, patients with fatty acid oxidation disorders are going to show manifestations of it, especially in the heart, the liver, the muscles, as you can see, an enlarged heart, heart failure, hypoglycemia or low blood sugar, low blood glucose levels, and then muscle weakness. On the right, you're going to see specific manifestations, and I mentioned earlier CPT1 and CPT2, and those have to do with carnitine palmitoyltransferases. And so what you're going to see there is you're going to find a fasting intolerance, hypoketotic, different seizures, or affection, the effect of it in the muscles. For all of these, and we're going to come back to talk about CPT1 and CPT2 in just a moment. Now, there's a diagnosis, hopefully there's a treatment, and thankfully there is. So there are 2 different routes of treatment, basically. The first is to counter hypoglycemia and the associated illnesses that come with it. So blood glucose is treated in neonates and children via an intravenous bolus of dextrose or glucose, it's really, really important that especially the infants not be put into any sort of metabolic stress through fasting because they can easily slip into a coma. So that's why you see the intravenous fluids. The second form of treatment is really stopping excess fatty acid oxidation. That's going to be preventing hypoglycemia through metabolic stress. That's has to do with fasting again, so you're going to need frequent feeding. You're going to be looking at low-fat and high-carb diets, and then for certain oxidation disorders, you're going to see carnitine maybe deficiencies, and in that case, you can have a supplement, a carnitine supplement. So carnitine is the main molecule that we're going to be looking at today, and when we talk about carnitine palmitoyltransferase 1 and 2 disorders. So as a general overview, carnitine is derived from the amino acids lysine and methionine, and they're of the isomers of carnitine. L-carnitine is the only one that's biologically active. So that for the rest of the presentation, when I refer to carnitine, I'll be referring to L-carnitine. Carnitine is essential for transport of long-chain fatty acids into the mitochondria for oxidation, as I've said, but interestingly enough, it's important for the transport of toxins out of mitochondria. I did not realize that until doing this presentation. So that's a really cool fact. Carnitine is produced in the liver, kidneys, and brain, but it's going to be concentrated in your skeletal and cardiac muscles. We can obtain a lot of carnitine through diet, looking at especially meat. However, that's not our main source of carnitine. It's not even necessary. You'll see vegetarians don't need to take a carnitine supplement because the body makes plenty of carnitine on its own. In fact, 95% of it is reabsorbed in the kidneys, and the rest is just going to be excreted in the urine. So carnitine deficiency is going to occur, but what we're going to be talking about is the transferase defect. So there are two forms, like I said, CPT1 and CPT2. Now, defects in CPT1 are going to result in a decreased transport of those fatty acids into the mitochondria. In other words, those long-chain fatty acids aren't even going to cross the outer mitochondrial membrane. These mutations can occur in the gene CPT1A, and that's going to mean that the carnitine palmitoyltransferase 1A is inhibited. So carnitine is not even going to be attached to the fatty acid. But then in CPT2, in carnitine palmitoyltransferase 2, defects there are going to result in carnitine remaining on the fatty acid and the fatty acid being unavailable for oxidation in the mitochondria. It also occurs because of a mutation on a specific gene, actually the CPT2 gene, and that mutation is going to inhibit carnitine palmitoyltransferase 2, which removes carnitine from the fatty acid, and so that the CoA can be reattached to the fatty acid because that's the only form that can undergo, obviously, beta oxidation. That goes without saying. So the physical manifestations or the consequences of these particular disorders has to do with a buildup of fatty acids in the liver, the heart, and the brain. There's going to be reduced energy, obviously, because you're not oxidizing your fatty acids to produce ATP, and your body's going to be in a hypoglycemic state, so low blood glucose. Like I said, these physical manifestations are the same in both disorders. The only difference is in CPT2, you're also going to have myalgia, which is muscle pain. So during this research, I discovered that carnitine defects can impact type 2 diabetes. So what happens with carnitine and type 2 diabetes is there's a reduced transport of fatty acids into the mitochondria, which means that there's going to be a buildup. It's kind of like a backup in the system. And that means that fatty acids are going to be backed up into the cytosol. And that is linked to type 2 diabetes. And it causes insulin resistance. And there's a couple different pathways by which insulin resistance can occur. The first is a defect in insulin resistance due to acyl-CoA and diacylglycerol accumulation. Um, then there's also mitochondrial stress and insulin resistance due to non-metabolized fatty acids. So, in other words, those fatty acids are either not ever even getting across the outer mitochondrial membrane, or even if they are, they're not able to pass into the inner mitochondrial membrane where they can be oxidized. Research has demonstrated that carnitine plasma levels are a good deal lower, actually 25% lower, in type 2 diabetic patients. However, they can be treated with an infusion of carnitine, and that has been shown to increase the glucose oxidation and glycogen storage. So as you can see, as you know, beta oxidation pathway is long, it's complex. There are a lot of different ways in which glitches can occur, but the two that interested me the most had to do with the palmitoyltransferase and looking at carnitine. And I believe that it's actually quite relevant considering how prevalent type 2 diabetes is in our society. And hopefully this can lead to different treatments for type 2 diabetes, which is going to allow people to have a much higher quality of life. Here is we're excited, it's pretty lengthy, but thank you so much for listening.